Hello everyone. Today I would like to talk about how to conduct 2D-QSAR model from preparation to publication. This is the outline I will talk today. The first I will mention about background about QSAR. Secondly, I will uh, discuss step by step for performing a 2D-QSAR model. Third is how to publish the paper, and third we can discuss um, uh, and I summarize discuss information about 2D-QSAR model. Let's talking about background. QSAR model is stand for quantitative structure activity relationship models, regression or classification model. As you can see here, you have data from the electronic database, lab collections, or literature data, and after that you combine to make the data set like that. After that, you can use the predicted QSAR float. Uh, in here, you can use several model. For example, linear regression model or non-linear regression model, ANN or Bayesian model to predict um, the PAC50 or EC50 of the compounds. And after that, you can select the best compound with high IC50. Finally, you can validate your compounds uh, with in silico in vivo to confirm your promising compounds. Next one is this is the slide. So we have around 6 types of QSAR model from 1 to 6. In here, I will focus on the um, uh, 2D QSAR model structure patterns with IC activity. This is the table, so a lot of software we can perform 2D QSAR model. Especially in here, in the number one here, we have the R software interface, and we can conduct a lot of model. For example, random forest, Bayesian model, artificial neuronal network, interface software, and you can download and you can perform QSAR model. Another software also free, but they need uh, your academic accounts, or they also give you around 2 weeks or 3 weeks for free. This is the graph, so the number of article related to quiz study increase in recent year. Compare with in 1971, I have a few paper, right? But during around 2023, the number of paper increased significantly, especially during that times, maybe the COVID-19, people stay at room, stay at home because this is the rules for the COVID-19 isolation. So a lot of people conduct the quiz study. So that's why the number of paper related to quiz study and COVID-19 increased significantly. Uh, because a lot of thousand paper related to QuizApp. So that's why they need the guidelines to make sure that the QuizApp model are reliable and can predict the compounds in safe and effective manner. For example, here the guideline 150, they require that we the data need the training set and test set, and also applicability domain to make sure that your model is reliable. Another guideline is, for example, 134 mention about skin sensitization, or another guideline for bioreadability or acute aquatic toxicity or repeated dose toxicity. Next one is the flowchart, how to perform the QSAR model. The first one is you have the list of compound, right? After that, you need to calculate molecular descriptors, and after that, you can run QSAR model. So after you run, you can make the correlation between uh, the actual activities and predicted activities. So the next step is you need to validate your model, and you can find the new compound with high activity for the uh, next analysis. So I will talk in detail about step one. In the step one here, you need to identify the Q chemical datasets. In here, you need to make sure that your compound are the same group. For example, your compound belong to alkaloids, flavonoids, or derivatives of drug or compounds. The compound must be have the same bioactivity, for example, anti-cancer, anti-diabetes, anti-Alzheimer disease, and they must make sure that they need to include IC50 or EC50. IC50, it means that inhibition concentration at 50% concentration, or if efficacy concentration at 50%. Uh, for example, here in this paper, they published in the he Haley Young journal, and they identified the association between the liquidity genin derivative and uh, alpha alpha-thin choline esterase, the enzyme to degrade the important roles in the Alzheimer disease. And you can see here the structure have contain around 3 benzene ring, 1, 2, 3. They also change the R groups in the compounds. So for example here, this is the structure of liquidity genin derivatives. The first structure, the first structure 3A and 3B only add more fluor in the benzene ring, similar with another structure. And then here they also include the PAC50. Uh, so how we can identify the potential compound, right? In here, I have several keywords. You can use the PubMed, this is the free one, with advanced, function. And here you need, when you click the advanced, they show you the table of that. And in here you need to put, the terms. In here I have, the first time I have the term is IC50, because I want to identify the compound they have inhibition of acetylcholine esterase. So I have the IC50, I put in here, and after that I click N, right? And the next one is I search similar with anti Alzheimer's, and I click N. Next I will find the compound with the synthesis or design. I also enter here and I click N. And the next one I will start with the review papers, because I want to have more compounds, so I want to include the review papers. So I add in the box here and I click end. Then finally I have the whole keywords here. So after I search, and then I find the paper they mention about the heterocircle compounds. They can against acetylcholine esterase and deuterium-choline esterase. This is very important enzyme to prevent Alzheimer's disease. In this paper, I found that they have around 311 compounds it belong to the heterocircle compounds, and they also include acetylcholine esterase and butyrylcherin turin esterase. So the next one I also did the same, and I find the article about synthesis of anti-cancer with the Eri Sunni Harazais against the breast cancer. And you can see here in the table 1 here, they show around several compounds, and they also show IC50 on different cells related to the breast cancer. The next one is easy one. You can use the database called GMBL here. The database including around 2.5 million compounds, and here they can link between your compounds and IC50. When you enter this database, you can only write down your compound purpose. In here, I refer to focus on the donepezil derivative, If I try here and I found around 208 targets, and in here you can select the whole compound here. So after that you roll down and you can see their bioactivity summary. In here you click the graphs and they can show you the IC50 of your compound. For example here, I click the graph and they show that donepezil in that case 20 nanogram, and you can click this link and they can show you the paper they provide IC50 and which cell or which animal they use to perform the test. And next one I will show you the my data. I did the chemical structure of anti-cancer in my study. I have around 230 phenolene amino pyrimidine urea derivatives. They can have the bioactivity like the anti-cancer agent. In here you can see this is the structure of them. They have around 4 benzene ring connected, and they also chain the R and H. R and H here you can see S and O and R here, for example, hydrogen between ethene, and also must be include IC50, and I mentioned before, right? Because it is the outcome. So in here I because my average of IC50 around 10, the 7.2, and the median of I'm sorry, it is median not mean, median of compound around 10. It means that my median is higher than mean, so my data was negatively skewed. So that's why I need to convert to the normal skew to make sure that my data can my model can predict perfectly. So in here, if you want to convert from the IC50 to PIC50, you need to minus logs of IC50. Next one, we need to identify the molecular descriptors. So what is the molecular descriptors? Molecular descriptors is defined for the final result of local, logical, and mathematical procedure. Uh, and in here, for example, 1D descriptor, you can calculate for molecular weight or element composition. 2D, including topological PSA or number hydrogen bond atoms, number hydrogen bond donor, or another indicators. 3D decryptors, including polar surface area, molecular surface area, molecular volumes, and uh, another decryptor sets. Next one is we have around 2,000 molecular decryptor can calculate by the machine, by computers, including 1D, 2D, and 3D. In 1D, we have at least 30, molecular descriptors. I will show you detail about 1D. As you can see here, in 1D, we have molecular weight, hydrogen atom, halogen atoms, hetero atom, until 30 molecular descriptors. So in here, I will show you how which software or which program can calculate molecular descriptors. We can use the GMS in the pre-software, and you can use Python, use the Mores web I use. You can use QSAR toolbox, or you can use the Gaussian, or you can use the Dragon to calculate the molecular descriptors. So today I will show you use the GMS. This is the pre-software. If you click the Chemdesk here, you have a lot of libraries. First is Padels, Chemopy, AdiKit, and CKD or Blue Desk. But Chemopy, they contain the whole, molecular descriptor, and you can. I recommend you use Chemopy to calculate maybe around 1,800 molecular descriptors. So next one is if you have the compounds, the compound I showed you before, right? and you use this software, and you try in the Google, and you can find this software, and you can convert your compounds from the image to the canonical smile in this here. And from smile here, you only enter in the input smile here to get molecular descriptors, and after you submit, you can download your data by CSV or Excel file. Next one is very important, how to select the best molecular descriptors, because we cannot use 2000 molecular descriptors. We need to select the best one. In here we have the rules. We need to select with the ratio 10:1. It means that you have 100 compounds, maximum molecular descriptor is only 10. And this is the process how to remove how to select or remove select the best model or remove the redundancy molecular descriptors. First, we need to remove descriptor with content values, content only content variables, or reduce the standard deviation less than over 1, reduce the missing values, and reduce the pair correlation larger than 90%, because if the compounds have higher correlation, they make the result distort. If if we can use a lot of methods to select the best model. For example, if you choose the stepwise regression, you can after that you can find the frequency of molecular descriptors. In here you only choose the highest frequency of the molecular descriptors for your model, because as I mentioned before, right, if you have 10 compounds, you only have one molecular descriptor. So depending on your data, you can choose the molecular descriptor with the highest frequency, as mentioned in the graph here. Another one, we use the Bayesian model average. In the Bayesian model average, you can select they can show you a lot of models here, and you can select the molecular descriptors depend on your data, right? For example, the model 1 is the best model. They show you around 6 molecular descriptors. The blue one here representative for negative association, and the rest one here so the positive association. After you select the best molecular descriptor, you can calculate, you can show your final data from right here. You have around here I show around 31 compounds with around 15 molecular descriptor, and here is my outcome. It is the IC50 bioactivity of my compounds. After that, you need to split data into trend set and test sets. In here, you can use a lot of methods. For example, principal component analysis or k-means cluster. And you can use ARM, ICTAS, or Stata, or every software. In here, for example, here after you split your data, you can see here the blue one present for test set, and the red one present for trend set. Or if you use the k-mean clusters, you can separate your data into two clusters in the figure here. The next one is why we need to separate your data into the trend set and test set, because the trend set would use to develop the QSAR model, and the test set to evaluate the effectiveness of the developed model. So here, after I run this one, and I will separate my data into the trend set and test set. For example, here the compound 16, 17 belong to the test set, and another compound belong to the trend set. After you split the data, you will go to the build the QSAR model. In this step, you can use a lot of model, for example, multilinear regression, multi-nonlinear regression, artificial neural network, Bayesian model average, and partial least square. In here, because each model they have their own advantage and disadvantage. So that's why we need to perform simultaneously uh, several models. So that, for example, here ANN here they have advantage, they perform well on complex data, but they have limitation on low interpretability. Um, so this is the graph I compare the predicted PAC50 and observed this from the lab, the original data from the lab, IC50 and predicted. And you can see here most of the model they predict so well, right? And you can see we have the uh, the strong correlation between the observed and predicted IC50. But you know, compared with other model, the ANN model, artificial network model here, so the perform superior compared with not the one. So DT model is the best one for um, uh, perform the QSAR model. This is the now I'm talking about detail about multilinear regression. In here, the first one you need to have the formula of multilinear regression. Uh, in my case, you know, I find that you know my IPAC50 equal around 4 plus with you know ET and plus with you know the dipole moment and plus with your log P. If you want to increase the IPAC50, you need to you know increase the DM, log P, and AT. In here, you know, after you run this model, you will show they will show you this is your data from experiment, and this is your predicted model from you know your model. In here I show the multilinear regression and ANN model. And after you have data, you can draw the graph like that. And in here, if you did the wet template and you use the BCA assay, and it is similar with BCA assay because you have the standard protein, and you from there you can predict how many concentration in your sample. uh, Next one is I will talking about step 5 validation of QSAR model. This is very important step. In here you need to calculate the internal and external. Internal, it means that you identify a lot of indicators, for example, R, IMS, ECF, or QR loop, or another indicators in the training set. And external validation, you need to identify a lot of new indicators here in the test set. And in here they also show you the expected and recommended results, how your model can pass this test. For example, here R square must be more than 60% and RC SEC is smallest possible, or something you can preference using this table. And the next one is you need to control the robust of model using Y-randomization. In this test, you know, if the randomization more than 0.5, it means that your test is passed. In here, I conduct the Y-randomization model, right? As you can see, this is original. I take from the multilinear regression, and I random around 10 model, and you can see here all of you know R square and Q in the random model is less than compared with original model. It means that my model is have a good internal reliability and avoid a chain correlation. So next one, I need to, as I mentioned before, right, I need to calculate the some indicator in the trend set and test set to validate my model. And you can see here I can in the training set I calculate around 5 indicators, and in test set I calculate around 3 indicators. And compare with thresholds here, my model pass the thresholds, it means that my model are statistically significant or statistically acceptable, or my model is so high internal external predict abilities. Next, you need to check the outliers because some compound they have higher or lower IC50 compared with the common one. And here you need to use the William plots conducted by the partial least square model. In here I found that to have two compound 20 and compound 26 here. They outline the square the square box here. As you can see here, we can explain that two compound here may be wrong experience data or wrong pictures in my compound, so that's why I need to include the compound in my data set. Next one, I will show you the step 6, visualize design ligands. Because we have 230 compounds, we cannot identify the whole compound, so that's why here we only select around 5 or 10 the highest PAC50 in the molecular in the your data set. For example, here the highest PAC50 around 6. So from here I will take from 6 to around under 6, around 5 compounds. And one I will try to molecular docking to identify which one have the high binding affinity with target protein. In this case, I will select the protein casing kinase 2 alpha subunit equal KCK2. It is the key player in cancer biology. It is the target for anti-cancer. And here, after that, I dock. Try to dock, the compound number 10, as I mentioned before, right? Number 10, number 3, number 6, 29, and 18 with the CK2, and I found that the compound number 10 here have the highest docking score, because in this case the actual number of docking is around minus the minus values, but in here I invert the docking score to make sure that we can easily to visualize. And in here you can see the compound number 10 you have the highest docking score, so I will select the compound for further analysis. After you dock, you can see that the compound number 10 here, they can bind with the CK2 with a lot of interaction, right? For example, the more potent one is the hydrogen bond with serine at the serine 51 and arginine at 47. And they have a lot of interaction, including one they work pi sigma, pi tax, amine pi tax, and pi archide. Next one we move to the stage 7, is the prediction and selection the best ligand. As I mentioned before, right, the compounds number 10 here have the highest docking score and highest not the highest, because here is higher than the compound 10, but and high PAC50, so we will select him for further. This is the compound of candidate compounds. This is the compound number 10, including the S. X here include S and ethans, and R here include 1,2,5 trimethylene 3 nitrocyclohexane and plus 2N. And here we have around 3 molecular descriptors, as I mentioned before, in the linear regression model. And here is the IC50, and we also identify the hydrogen bond. Next one is identify to check whether or not the compound met the least spin rule or not. Least spin rule including five rules, right? Molecule weight less than 500 grams per mole, log P less than 5, the hydrogen bond donor or hydrogen bond acceptor, less than 10 or 5, rotation bond less than 10, and the PSA less than 140 Armstrong. And in here, most the compound number 10, met these pinky rules, and they consider oral active compound. And next one, as you can see, this is the structure of compound number 10 here. The pink one it indicate that the compound belong to the optimal range, and the compound number 10 here, as mentioned in the figure here, they belong to the optimal range. It means that your compound number 10 is orally bioavailable. And as you can see here, the compound number 10 here, they can cross the human intestinal, and also cross the blood-brain barriers in the organs, shape here. And the more important one is the compound number 10 is non glycoprotein. It means that when they enter the brain and the brain did not remove the compound, it is very popular compound to treat the cancer in the brain. Next one, we check the ADMT. It means that the absorption, distribution, metabolism, excretion, and toxicity. As you can see here, as I mentioned before, right, the compound number 10, they can absorb in the intestinal. They can they pass the Charcot-2 permeability, and they need the water solubility, and they can cross the blood-brain barrier. Here, compound 10, they act two options: subtract and inhibition of several enzymes, including so it means that because it is a very important enzyme in the phase 1 metabolism, so it means that the compound may be interact uh, with not the drug, so very important when we use the compound for treat cancer. And the next one is we identify the existence of renal substrates, and a more important one is the compound we have no toxic, including MS toxicity and skin sensitization. Next one, we detect the bioactivities, as I mentioned before, right? If you have the smiles, and here you enter in the path to online here, and you can identify the bioactivities. For in this case, I find that the compound number 10 here they have around 80% they have the activity um, uh, for the anti-cancer, around 60% for antioxidant, and around 13% for anti-inflammation. And so this is very similar with previous study. It means that the compounds number 10 is predictable. Next one is we need to validate the compound in experiment, right? Because if you don't have labs, you can use the compound to use molecular dynamics to validate your compounds. This method can replace experiment because some experiment difficult to perform, or too dangerous, or expensive, time consuming, or by too many parameters to control. So similar molecular simulation dynamic here, they can replace the experiment, they can provoke experiment, explain and experiment, and complete theory and experiments. Uh, Here I show you a lot of software to perform the molecular dynamic simulation, but you need to have a strong computer. For example, GOMAS, VMD, Amber, Schrodinger, or LAMS, or NAM, a lot of software. After you use the software, you can perform the video that, if you can see here, this is the compound number 10 after docking with CK2, and you will simulate it in the in the water environment. In this case, I simulate in around 100 nanoseconds. In that case, you can adjust for temperature, pH, pressure, salt, velocity, and everything similar in reality in cell and vivo model. And after you conduct that, you can see here this is the compound number 10, they call ligands, and this is the CK2 receptor. And during 100 nanoseconds, you can see the compound number 10 still stable and attached with protein. It means that the compounds are very potential for treated cancer. After you did that, you need to show your data with 4 figures. The first one the stability of the compound with protein with the IMSD. As you can see, during 100 nanosecond, the compound still stable, right? Little bit increase during 60 seconds, but the fluctuation, the variance is not too high. And it is the graph show the binding between the compound number 10 with amino acids. You can see here in some amino acids the fluctuation so high, but in this case did not affect the stability of the compound and protein. Here the hydrogen bond play important roles in the complex of protein and compound, and you can see from the 0 nanosecond and 100 nanosecond, the number of hydrogen bond average is 2. And the next one is we identify the stable of folding proteins during 100 nanosecond. The folding of protein is still stable, so that's why the compounds number 10 is stable when we simulate they're complex in water during 100 nanoseconds. I mean that the compound number 10 is very good, the promising candidate for treat cancer. So in here I will conclude my finding. Right, I have around 200 compounds. They have the activity to against cancer, right? But we don't know which is the best compound. So after that, I will use QSAR modeling with a lot of model, and after that I predict, and I will find the promising compound for anti-cancer, and it is the structure of compound to treat cancer. Mm, so after you repair your paper, and how you can publish your paper, right? And here I show you a lot of publisher you can use to publish your paper, including Elsevier, Springer, Taylor, Francis, William, Nature, Cambridge, or ACS. In here, in this journal, they have the keyword journal finder, and if you try the journal finder of Elsevier, Springer, or Wiley, or Taylor Francis, you can find. They can show you the the um the website like that, and after that you only enter your paper titles, paper abstract, and you find the journal. In here, after you uh, enter your data, and you can show you a lot of journal related to your paper, and you can publish in there. In here they have two types of publication: open access or subscription. Open access, it means that you need to pay money when you submit your paper. And for example here, this journal molecular structure here, they charge you around $3,000 for one paper. uh, and if you choose the subscription, it means that your paper is free. It means that you no need to pay. But when you want to read the paper, you need to pay around $30 or $50 to download your paper. Next one is another page journal is another one is they can show you the cycle of the paper journal, impact factor, acceptance rate, time to the first edition, and time to publication. Very detailed one, and you can choose the best journal related to your compound, to your papers. She is similar with another one, computational biology and chemistry. Next one is I will show you my YouTube channel. This is my YouTube channel, and here mm, uh, I make the whole video how detail how to perform step by step to perform the QSAR model, and also including bioactivity and molecular mechanism related to compounds. If you interested, you can come to my journal and you can read, and you can follow me, and you can um, repair, and you can submit your paper. Uh, please help me subscribe to encourage me make more video. Thank you for your listening. Thank you so much.